This is a commonly confused topic in cell biology. This is the difference between the rough endoplasmic reticulum and the smooth endoplasmic reticulum. And I've added the Golgi apparatus in there as well because it's another one that looks very similar to the other two, but again has a slightly different function. And if you find these explainer videos helpful, then please leave a like and you can follow the channel for more. Okay, so these will be found inside eukaryotic cells, and people often get the rough and smooth ER confused, but there's a very easy way of telling them apart, and that is the appearance of the ribosomes on the outside of the rough ER. So if we look closely, we can see all of these little yellow dots on the outside of the rough ER just here, and they are all ribosomes, and that's why we describe it as the rough ER, because they give it this rough-like appearance. And they also give us a clue towards the function of the rough ER, because ribosomes are used for making proteins, and the job of the rough ER is to synthesize and transport proteins, particularly those that are going to be either secreted from the cell or going to be sent to the cell membrane. Next we have the smooth ER, and we can also see this lingering in the background of this picture of the rough ER, and that's because the two are often attached, but they do have different functions. So the smooth ER is called the smooth ER because we can see there's no ribosomes on the outside of the smooth ER, so it doesn't have that rough appearance. And that's because the smooth ER isn't involved in the synthesis of proteins. Instead, it's involved in the synthesis of lipids and carbohydrates. And in some cells, it can also be involved in the detoxification of harmful substances. And then down here we have the Golgi apparatus, which has a slightly different function again. So after the proteins have been synthesized at the rough ER, they are then sent to the Golgi apparatus for packaging and transport. And sometimes it'll be involved in modification as well. For example, it might add a carbohydrate to a protein to make a glycoprotein. And then we can also see these vesicles budding off as well. And these vesicles are responsible for transporting the proteins to wherever it is they need to go. So in summary, the rough ER is for synthesizing proteins, the smooth ER is for synthesizing lipids and carbs, and then the Golgi apparatus is for packaging and transporting these products, and then sometimes modifying them when necessary. But they all work together as part of the cell's secretory pathway, which is like the production line, which is responsible for making and exporting the cell's products. And that's all you need to know. So thank you for watching, and please let me know what you would like me to cover in the next video.